as I said earlier, one of the most important topics for biology, and expect quite a few questions on that. And I was trying this morning to pick up the update for influenza. So basically, it's an influenza that's considered as one of the serious problems. And you can see, as I said the other day, WHO, CDC do maintain the world record. So you can see this is status as of 20th March 2015. So this morning, like 15 minutes ago, I just downloaded this statistical analysis from CDC. And you can see they talk about the percentage influenza positive as of today. So you will see the whole map of the world. That includes Canada, US, and rest of the world. There is a problem there. And again, we need to be fully aware of this statistical analysis for especially influenza. And I'm going to give you a little bit of historical significance so that you keep in mind why do we care for influenza. And then again, those of you who want to go for drug development, why there is so much energy going on. And you can see Tamiflu happens to be one of the drugs which is out there if such a thing happens. And I would encourage you, those of you who have problems reading textbooks, and they have problems even going through the PowerPoints, so they may find some of the public information. Remember, as a physician, as a pharmacist, there's always a challenge these days. There were good times. There were good times, I would say 10, 15 years ago, when the layman, the patient, did not know anything. And even if you read that particular subject in medical school or pharmacy school 10 years ago, you pretty much had good knowledge to overcome their problems. But these days, with all good thanks to Google, most of the patients and most of the people would know more than you, because that's the only thing they want to go and study whole night before they come and see you. And they're going to ask you questions. So our dealing with the patient is going to be different. We are living in a different world. And guess what? What kind of embarrassment you're going to go through when your patient says, yes, I saw CDC this morning, and they were talking about such and such drug. Is that drug available? And you're wondering, oh my goodness, I just read the class notes and those 350 questions that Dr. Fazal posted me. I have no clue at all. Well, so that's going to be a challenge for you. But anyway, the other important thing for you to know is what is the public information there. So for example, this is from CDC. They want to educate people, and you can see where do I stand in terms of virology understanding as compared to a normal person. So we already know the flu virus enters the cell, creates many copies of itself. This is public information for a high schooler or for a layman. And then again, neuraminidase is an enzyme on the surface of virus. This is an enzyme on the surface of virus, and the virus needs to break away from the cell in the last part of viral replication, which is budding. And Tamiflu, the best-selling drug, basically is a neuraminidase inhibitor. So that will be very unfortunate if we don't know what the action of Tamiflu is. I'm just giving an example. And the whole idea for Tamiflu is that it prevents virus copies from breaking free from infected cell to spread. Simple. And you can also see the producer rush, how much money they're going to make up, because it's all over the world. Just one drug, one person, and there you go. So let's begin with RNA virus and orthomyxovirus. Influenza, of course, I just picked up one of the important topics. I'm going to go through the general family characteristics of this virus to give you an idea, and then we'll go in detail. Now, all of us know orthomyxovirus, basically influenza is the major thing. And there are many different things happening when we talk of influenza. I just gave you the CDC, the WHO statistics loaded this morning. It would give you an idea that even now, as of today, influenza is there, right? So that kind of a presence of influenza at all times in the whole world, that is called pandemic. So you can see it's there, endemic. So it's already there at a given level. And then you get pandemics. So there's something on top of that. When you get surges, you get peaks. That's where the problem is. Historically, you can see from here, I'm going to give you some statistics as well. 1918 to 1919, 20 to 40 million people. So it's a very high incidence mortality in terms of loss of human life. And if you put things together, we basically lost more people because of influenza than the battles of World War I. And then again, the other important thing is a trend that these viruses have. So they keep on coming back and forth. We don't know why that happens. That's why there is something called, which is called emergency preparedness. As we talked about bioterrorism, we talked about the preparedness that government has, and they always take you to exercises. So you can imagine if things of that nature comes again, that we jeopardize the life of 40 million people. It's like 1/6 or 1/7 of the population of this continent. It's a lot of people. And you can see it came 1918, 1947, 57, 68. 1977 was the last pandemic where we had such a significant number of people dying. The good news is nothing has happened ever since. There were a little scares last couple of years. And the other problem is that the new virus strains have been developing. That's what I think I need you to understand. What's the problem with influenza? And that pretty much will answer questions that patients have. Why do we have to have flu vaccine every year? What is this vaccine different than the last year? Things of that nature. So I think that will be something that you want to take from today's lecture. Then again, also keep in mind that you can give people vaccines, but of course there's no such thing for you to take antiviral drug for prophylaxis. For example, you saw Tamiflu, the action is stops budding. Of course, virus has to infect you before you stop budding. So if you take Tamiflu without viral infection, there's no use, correct? Simple? Okay. Now, if you look at the timeline of death, and you can see there have been plague as one of the causes. We have had cholera. And of all those timeline of infectious diseases or some of the things that have killed humanity at large, other than the world wars, you will see that they have been pandemics for flus. We call like Spanish flu. We have a Hong Kong flu, Asian flu, so on and so forth. So that's like a timeline. The point I'm trying to convey is that for some reason, it comes back. So this means already is existent somewhere, but it comes back over and over again. The problem with this kind of thing is, I mean, just like imagine 10 people become sick, 20 people become sick, 1 million people become, where are we going to keep them? Who's going to look after them? Who's going to go and even touch them, especially, ethically, morally, and so on and so forth. But you can see from here, it's not only a challenge for taking care of the living one, there's going to be a challenge for taking care of the dead one as well. So that's a very, very serious concern for all health-related professionals. And you can see from here as well, if you look at global picture, 40 million in Spanish flu in Spain. Russian flu, we don't know. There were no statistics. But the other important thing I want you to pay attention is that they can go and figure out the strain type. Most of the strain type, they are talking about two important adhesion molecules, H and N. H stands for hemagglutinin, and N stands for neuraminidase, right? So it's going to give you a number H1, N1, H3, N2. So every new strain has to have that numerical number and assignment. So that's another important thing I would want you to pay attention. And for some reason, if you have a pandemic in the past where people were not able to serotype that would be again a challenge, because when something new comes up, we have to look back, what is it, where is it coming from? Again, the same message, you can see H1N1 from 1918 all the way through 2006. The latest one we had was H5N1. So 2006, we had H5N1. And that's what we have a problem, February 26, 2006, 92 deaths and 170 cases. So you can see on the top over here a cartoon of a virus where it has an H that jutting out or protruding out glycoprotein complex. And if you remember your basic immunology, that's the adhesion molecule the virus needs to attach and detach as well. Attaches for attachment and detach for budding. Again, H1N1 incidences, you can see we talked about H1N1, and then you saw in US 227 deaths, Canada. So all this, especially for this continent, is important because this had a major brunt of infection as reported by the national focal points. Okay. If you look at, for example, pandemic that we have H1N1 in 2009, again, we saw like 77,000 cases and there are 332 deaths. But again, you see predisposition of South America, North America, and Australia, and some of the European countries. So this is something where government has to chip in and put travel bans and things of that nature. Now, if you look at the family of orthomyxoviruses, they are basically A, B, and C. And the good news is that only influenza A and B cause human disease. So that's another message for you. And if you go back, look at the picture that I showed you a while ago, where we talked about the RNA and DNA differences. All you need to know is that the RNA is basically a negative sense RNA genome, and it's segmented. So if you see like a stair-like pattern over here. The other concept that I want you to understand is that why do viruses mutate? Why do they change? In genetic terms, we call it genetic instability. And that basically is the one that causes annual epidemics. So we have a new strain every year. So there are two important concepts that you have to understand. One is called a mutation. This is called drift. And the other one is called reassortment, which is called shift. And as I said, it will be good for you to know a little bit of genetics, and I'm going to give you an example for that. I usually give an example that if you are walking or driving on a highway, you want to make U-turn, that will be like a major shift. So there's something, a major turn of direction, as compared of you're driving on a highway and you're kind of a little bit sleepy or under the influence, the car goes like drift to one side to the other. So that's like a minor thing, correct? As compared to you. So keep that concept in mind, and I'll take you from here. Look at the virus. This is a model of influenza A virus. Again, you will see RNA in the middle with some of the enzymes that these viruses come up with. For example, polymerase. And then there is an outer matrix protein like a capsid. And then we have a bilipid membrane, as I said earlier. That's where it comes from the host. And the most important thing is Hemagglutinin and Neuraminidase. So you see tons of them protruding out like spikes, like attachment molecule. If you look at the electron micrograph, pretty much you will see the same picture. Okay. You can compare, and I think I'm going to emphasize more on A and B. You can see from influenza virus A and B on the left-hand side as compared to influenza virus C. You will see most of the stuff that we have for A and B is Hemagglutinin as compared to some other type of glycoproteins that are obvious in C type. And of course, if you have a saccharide chain or sialidase, depends upon what molecule you are looking at. And this is basically a drug development kind of a slide that basically, you remember I gave you an assignment to come up with, for your individual projects to come up with like drug and delivery. And basically, this particular topic of all the ASM people was, I think, given, is she here? Ayla, is she here? So most of the slides that you will see different from the slides that are posted are basically coming from her. So she posted some of the latest one, and I really think they're useful. Okay. Now, if you want to compare A and B, that's what I usually ask people. Which one is more dangerous as compared to A and B? And then again, what you need to keep in mind over and over again is, as I said, genetic instability. So you have H and N. H stands for hemagglutinin, and N stands for neuraminidase. And if you look at their functions, viruses need that to attach to cell membrane, right? Hemagglutinin. So keep in mind that agglutinin is something that basically what it means is the cells agglutinate, as you saw in those labs, that they coagulate, they come together, stick together. That's the word that you use, especially for the red blood cells. So it kind of brings things together. So that's what the attachment to the membrane is, as compared to neuraminidase, which gives you a little bit of a sense of an enzyme, ACE ending like an enzyme. So enzyme is like cutting, breaking apart. So in this case, it's going to be released from membrane. Okay? Now again, you can see from here on the top, hemagglutinin binds to the receptor on the host cell. So that's an important concept. And then again, a neuraminidase, it says, I just showed you release, but keep in mind that when virus releases, it's going to go and attack other cells. So that's more of a initiate infection. And then there are different kind of RNA molecules within the cells. Because remember, again concept-wise, though these molecules are expressed on the exterior surface, what is the message there? The message for transcription of these particular epitope is there in RNA. So RNA is where this is coming from. And then again, that RNA can come from different sources as well. So keep in mind, for example, you can see from here, it could be a swine origin. It can be of human and swine origin. It can be many different type of things that these viruses can go infect and pick up that particular nuclear material. So if the virus infects a swine, infects a human being, and the same virus is transferred, it's going to pick up a genetic material from one and pass it on to other. Okay? Now, again, a little bit more of hemagglutinin. As I said earlier, it's going to go and look for a polysaccharide on the cell surface and attach and then inject the viral genome into it. On the other hand, neuraminidase is a complete virus, is going to clip off and then move off from the cell. So that's the basic concept in terms of these two important molecules expressed in the surface of the cell. And for influenza C virus, as we discussed earlier as well, you will see that we have a specific, in this kind of a matrix around it. You can see a very specific matrix. And the other important thing is that hemagglutinin esterase fusion, that was not there in A and B. Right? So that will make a difference in terms of what is there on the outside. We're not really bothered for influenza C virus at this stage because it comes from the same family. So we just want to look at and study some of the molecules, because when we do experimental studies, we want to find out, and if I was to work in the lab, I would rather work in a lab which is less contagious and less infectious, but gives me pretty much the same molecule that I can play around and experiment and block. So that's where some of the stuff comes from. And again, keep in mind, though the RNA is there, but as I said earlier, many of the viruses will come with their own polymerases. They're not even dependent upon your cell's polymerase. Well, maybe they think that cell may not provide, those cells would provide it. so they can come up with their own machinery to go and help themselves to divide. Okay. Now, also keep in mind, let's pay attention to this one over here. And as I said over and over again, you need to understand the basic viral replication, and the rest of the system is easy to understand. You understand the concept and you can change the virus. So you can see from here, in this case, we have a virus which has a H molecule and an N molecule. As I said earlier, there is a polymerase complex within the nuclear material. You can see the polymerase complex within the nuclear material, and there's a viral RNA. I mean, these are a little bit sophisticated slides, and but I think without going in detail, concept-wise, they are good. And you can see it will give you an idea that why do we have different strains. For example, if you were to look at this polymerase complex, you can divide them into different groups in terms of what kind of H and what kind of N do we have. Remember, when we go for a new pandemic, we want to find out what is it there for H and N. So you can see from here, there are different choices that we have. We have a lipid envelope derived from host cell. We already talked about that. And also keep in mind, when I say host cell, and I also told me it could be bird, swine, and human being. So we can also differentiate if that virus is coming from that particular animal or particular host, right? So that will be another important thing. So let's say this is a cell over here. So it expresses a receptor, and this is a corresponding receptor. The first step, again, is going to be recognition of that cell, attachment of that cell, going into it like endocytosis. You can see endocytosis. And then again, it's going to break apart. We call it uncoating, and then release. And then the whole idea is that after fusion and uncoating, now virus is going to use that cell's machinery for macromolecular synthesis. It's going to break apart all these things, and with the messages will go to the nucleus. We want to make each and every component of that particular virus. So you can see all these sequence, and also keep in mind the transcription of protein synthesis is in messenger RNA. So there's a message there, a code. You can see different color, different configuration of the color. It's going to send it, this information, from here to the nucleus in a negative sense RNA. And then nucleus will start transcribing. So it's going to be transcription, translation, initiation of protein synthesis, and we have ribosomal, and then we have Golgi apparatus, and then finally we want to make multiple copies of these sequences, and we do what we call post-translation processing, and then again everything will be complete. Now this virus has to pick up the outer covering bilipid of this particular cell. At the same time, it's going to send a message that we need to make those specific H and N proteins. So you can see on the top, the H and N proteins will be assembled, and the virus will bud off. And the question, why do we need to know that? We need to know that because we want to stop that at any level. We're going to use all different drugs. But also keep in mind that Tamiflu, for example, is basically going into that game a little bit late, isn't it? So we already have allowed viruses to attack and this and that, and we're just playing the game in the end. That may not be a good game to play, right? You just want to stop the person scoring a touchdown right near the penalty point or whatever. It's too late, so it's hardly, but it works. Okay? So you can see from here all different components based upon your knowledge of a little bit of biochemistry, a little bit of protein synthesis, because these are by the end of the glycoproteins. Okay? Now, what is it that determines that you will get a disease or you would not get a disease? Okay? Then again, keep in mind two important things. One are the viral factors on the top, and then you have the host factors. So that's the game plan here. So virus is coming, this is a typical virus. Virus is coming from many different things. You can see from here, for example, virus says that it's bringing in a growth and a bacterial growth or antagonizer and interferon antagonist. So remember, interferon, when you get viral infection, you immediately release interferon gamma. So that will activate or push up, kick up your immune system. So virus is coming up to stop that. Very intelligent virus. So you can see that's over there. Then again, virus is coming through receptor binding, antigenicity and tropism depending upon the presence or absence of. So it's coming with all the sequence of virulence agent transcribed into it, ready to go and infect. So virus is lethal in a way, and it can be even severe. And the whole message to the virus is that virus needs to be keeping all those enzymes if needed. It needs to attach, it needs to multiply. So all those things are within the viral genome, so to speak. Okay? Now what do we have on the other hand? So if you look at the other hand, for example, host factors. What is it that's going to determine who is going to get that virus, who's going to get fatal disease? As you can see, if you are more than 65, less than 2, pregnant women, immunocompromised, that pretty much hold the situation for all viruses. But again, this is the virus. If you have underlying health conditions that will increase susceptibility, asthmatic, chronic obstructive air disease, cardiovascular disease, obesity, diabetes. And then again, many other things which are required in terms of you fighting that viral infection. So you can see from here, things are not simple. When we in our simple minds, we think, you got flu, I'm going to give you Tamiflu. Okay? But as I said earlier, the whole concept of medicine is changing. And we're talking about personalized medicine. We're talking about genetic medicines. We're talking about every gene is different. Every person comes with different kind of genetic background, different ability to fight disease. So we may have very specific drugs that are going to help one person more than the other person. Okay? So that's where it is coming from, and you can see that there are other host factors that influence viral infection by either direct or indirect interaction of the viruses. So these are some specific things that we want to know. For example, your ability to produce gamma interferon, your T cell's ability to act in time, and then again, if you don't have those compromising situations that you saw in the host factor. Okay? Also keep in mind, and this gives an example of a virus versus human host, but it could be some other thing as well. So in this case, you can see it could be swine, for example, or it could be a bird. Because we human beings are living in an ecosystem that we come and interact directly or indirectly. You know, I was interested in finding about the avian viruses and where it started, and it was a town in China. And then I looked at the market, and they were like open market with all these birds and this and that, and you know, their waste and excreta open in the market. So you can look at the pollution over there. And the people are going to go and buy something and take a flight next night or next night and then fly all the way over here. Right? So you can see how fast things can be transferred. So the global influences of all these factors are also important. One thing. The other thing is, the question we may ask is that the virus, is it going to be the same virus that has affected swine virus, for example, and is going to affect human as well as compared to the bird? So you will see in this whole cycle, the basic virus is the same. So you have a basic virus with basic, machinery and basic pathogenesis factor. And the question you have to ask is that can a human virus infect a swine, or a swine virus can infect a human? So you will see over here, especially, there is an overlap. There is an overlap. For example, if you remember, especially, I think it was Mexico, Brazil, I forgot. So people working in the farms, and just imagine who's going to care for those animals, whether they are infected, who's going to go and check? I mean, there should be. I'm pretty sure there will be over here to check the health condition of those animals before you slaughter them. But just imagine if by chance that system fails. And meat is there, and that meat comes and you consume and so on and so forth. Or people who come in contact with those people, they are the same people, they move around. With this modern technology and aviation system, anything can travel within hours all over the globe. So what the scientists are doing is that they are looking at, for example, a virus that goes and infects a swine. And of course, it's going to go to the swine cell, pick up a nuclear material from the swine cell, and then pick up some of the nuclear material from human cell. So it's going to be mix and match. So that nuclear material is going to be mix and match. So that may happen into these viruses, and that's what they go and study and find out that if there is going to be any change in terms of expression of those specific antigen molecules. And then again, you can see over here, some of the viruses, for example, may have receptors that will go and attach to your upper respiratory tract. Some of the viruses may have receptors that will attach your lower respiratory tract. And you can also see some of the viruses want to be at a specific temperature. In pigs, for example, 39 degrees C as compared to human. And then again, we have avian viruses as well. So viruses per se don't really care as long as they can get cells. So these days we may have a mix and match of different colors of genes and so on and so forth. So if you were to look at the genetic analysis of these viruses, and you can see on the top, humans, for example, 1918 we have H1N1, right? When we look at the classical swine as well, which is also H1N1. So you can see kind of similarity. Then again, you can see over here North American avian, then human. And you will see mix and match of everything. So all different kinds of proteins are exchanged, and then you can come up with any weird kind of viruses in this case, which can pick up those specific messages. So you can understand the complexity of the problem, number one. And also remember the ecosystem where we are living in, and also remember the need for us to study. Because they have to wait till there's an outbreak of virus, type this virus. I'm going to talk about it like in the last few minutes. How long does it take for them to prepare for vaccine? It's not like right away. It takes time. So everything is time consuming. But you can see from here similarities and dissimilarity, and where these hemagglutinins are coming from, where the viral segments are coming from, and what are their specific needs in terms of the need for them to replicate. This is basically a legend of the previous figure, but the most important thing is that right now there's no vaccine against influenza C virus, but again, I did not pick up influenza virus C because it's just going to complicate. I think for you, for this class again in AB is good enough. This is, I think, a video, as most of my ASM people actually, when they were working on this, updating my lecture slide, I told them to introduce videos, and you will find videos for most of the people. I think when Ayla was working on that, she put that video as quite useful. I'm not going to play it, so I would want you to play on your own time. Now, once you get a viral infection, it's a classical flu-like symptoms. So you had fever, malaise, headache, myalgia, body. There's no difference than anything else, as we discussed in the common flu as well. The difference is the nature of virus. And as I said the other day, we say stomach flu, intestinal flu. By all means, flu has become a layman's term, misappropriately used. Flu should be reserved for influenza, especially influenza A. You can see the symptoms, especially for the swine flu. When there was an outbreak of swine flu, this is from National Health, and you can compare that. But I think I would suggest it's like a vague thing. Now let's do the A and B together, like some of the features that are common. And the common features are that they are enveloped virion. They have 8 unique negative sense RNA. This is one of my favorite questions that I usually ask. I would never ask you the negative sense RNA nucleocapsid segments. And then again, this part, especially for some of the drugs that we use for viruses, for example, amantadine and rimantadine, and again, you can go back to the previous slides. You have to keep in mind that these two are acting on the uncoated step. And this would probably mean that if you don't get Tamiflu and you have a reserve for amantadine-like groups, you can give it to influenza A patients. Okay? Whereas if you have the other drugs like antiviral, zanamivir or oseltamivir, these are the same Tamiflu-like drugs. So they basically inhibit the neuraminidase protein, and they can be used for both A and B. So that's the difference between two lines of drugs. And then again, we discussed earlier, influenza A is the dangerous one. The reason being it affects human, mammals, and birds. And anything that affects birds or animal is called zoonosis, from zoo. So zoonotic diseases, something to do with zoo. Okay? So I think the upshot of what I told you is that there are M2 inhibitors only used for influenza A, and there are NA inhibitors. They are used for A and B as well. The other important thing is, well, they recently are changing Tamiflu. You can have in other forms as well, but most of the time the availability is oral as compared to zanamivir, which is inhaler as well, because of the fast action that you may have. It will be good for you to kind of get the basic understanding in terms of the antiviral drugs, and you can see it's going to come over and over again, but I want you to maybe those of you who learn by drawing things, you can put all those viruses that we have discussed so far, have one cell, and see what are the differences between all those 8 and 9 steps, initial steps and later steps. For example, if you were to look at this one, again, you can see binding number 1, number 2 is uncoating, and then again, number 3 is that negative sense RNA goes into the nucleus, transcription takes place, translation takes place, protein synthesis happen, all those different components which are there in terms of sending a message for making those specific receptors are made, and then endoplasmic reticulum, everything is assembled and then bud off. The most important thing that you will see over here is all the components of the viruses are made within cytoplasm and nucleus except the important one that we want to block, hemagglutinin and neuraminidase, that basically are expressed on the cells before the cell buds off. Okay, so that will be the primary theme for all viruses. The only difference in this one, as you would see over here, is the negative sense RNA segmented of RNA, and then again, these are those receptors that are made. And again, as I said earlier, we are trying to develop, and we want to make sure that we can go and develop some of the drugs that can basically block the expression, but also keep in mind some of the drugs may be general. Okay, this next one is basically a legend for the previous figure, and if you, those of you who are visual learners, they can read it up. It's pretty much what I just described. Now the basic concept, I'm going to go for two concepts today, antigenic drift and antigenic shift. So these are the two major things. I just give an example, you're sleeping, you're just drifting. It's kind of a minor challenge as compared to you making a U-turn. So keep that analogy in your mind. Antigenic drift by all means is a minor antigenic change. And basically it results from mutation of H and HA and NA genes, and this is called antigenic drift. This process takes 2 to 3 years, and this is responsible for the local outbreak of both influenza A and B. Okay, now you will wonder, it's too detailed. It's too detailed. Maybe it's nothing for me to know. Also keep in mind the public information. You can imagine many things are there as a public information. So I don't know who goes and decides that. But the question that if you are there in a pharmacy, say, why do you have to get this flu vaccine every year? What is the difference, so and so forth? So you can see that, for example, on the top it says that when you are infected with a when you're given a virus, they basically prepare those specific antibodies known to them are expressed based upon the antigens of that prevalent virus. For example, it has, let me give H20, N20. So they make antibodies to H20, N20, give it to you. You are prepared to take that challenge. The moment that virus comes over here, you already have this antibody, and H1 is for attachment. Antibody will come and block that. So that's where the flu vaccine is. Does that make sense? Okay? And then, for example, you can have tons of these antibodies. And these antibodies are designed. So I had a flu shot last year. So I have the antibody for that particular flu that was prevalent at that time. And I may still have those antibody, but now the virus has come up with another antigen. And this antibody would not bind to this antigen because it's non-specific. Okay. It may, but it is not designed for that. You can see viruses have this ability based upon, they will still have the same hemagglutinin, same neuraminidase with a different epitope. And the viruses, the antibodies are very specific. So this is basically public information to tell people that's what happens. They need to have that, and this is based upon a scientific fact. This concept, where this changes in terms of antigenic and epitopic nature, is coming from a genetic thing which is called antigenic drift. Okay? So far, so good? Okay. Now, the other concept is shift. I give an example, U-turn. How do you make a U-turn? Is a big challenge. This is a major change. This is a major change. And this is coming from reassortment of genomes among different strains. That's where mix and match taking place between humans and animals. That's where now the problem is coming. And this only occurs with influenza A virus. So what it's going to suggest to you is A virus is more lethal as compared to B virus. And that's where the problem was. The previous one may have some kind of endemics going like at a fixed level, not to worry about, but pandemic is something that's going to involve a panic, right? So pan for a panic, it should create a panic because it's going to go after millions of people. So that's what we are caring for. Again, public information for people to know. You can see from here, number one over there, that he's talking about two different animals. So you're talking about swine flu, you're talking about the bird flu. So both get affected with the viruses, and then the viruses also influence human beings. So whichever host they go, they pick up that kind of machinery over there. So it's going to be mix and match. So you can see from here, there are three different viruses. They will have three different kind of the structural element. And if you happen to be a person, for example, like this is a human being, and you get your own virus as well, and you get a bird virus as well, and both of them are there, and they are going to share many genetic things, and then you will come up with a new strain altogether. Okay, so that's basically is a very severe form of disease. We call it a killer flu mutant. So that's where the major problem is. Again, I picked up, not me, but those people working for me for ASM, they helped me out to do that. But it's good to get a feedback, because sometimes those of you who are visual, they may kind of register and look at these slides over and over again and form concepts. And then you can see from here, as I said, there could be different methods. There could be different methods of causing antigenic shift. So you can see the bird has a virus, and this bird influenza A virus happens to go in human, and the human then passes on to 3 humans, and 3 humans are genetically different, and this goes wild. That's one of the methods of reassortment of gene. Number 2 is the bird infects swine. And the swine, the pig comes in contact with human being, and then human being passes on to the other people. That's the second method of the problem. The third one is even a severe form, where a bird and a human goes into swine, and swine does mix and match, and then comes back to human being. So you can see the complexities of this situation. And of course, in all these situations, you will see by the end of the day is going to cause a pandemic because human beings are going to come in contact with human beings. So one person working in a farm in a South American country get infected from his swine, traveling all the way over here, passing it on to the rest of the people, and then goes on and on in terms of a viral replication. Again, the same concept in terms of the specific nature of that molecule. So you can see, like a bird, it has H5N5. Human beings have H3N2. They both co-infect swine, and swine does reassortment of these different genetic elements, and you come up with a news, and it goes to the human. So then you want to trace back and find out that where is that particular genetic thing coming from. And again, typical pathogenesis, you have an aerosol inoculation, goes to your respiratory system. It goes to your, depends upon whether you have an antibody in your system or not. You have a good T-cell response. Pretty much the same, regardless of which way you want you to look at, but eventually you will have influenza syndrome. And then again, you may have pneumonia, and many times when you have viral infection, you may have secondary bacterial infection because your immune system has gone down. That happens many times. People say, okay, don't take antibiotic because you just have a common cold and flu. That's true. But over time, you may end up taking an antibiotics because you get secondary infection. So this is where it comes from. And you can see, the whole idea is if you have a good immune system or you're protected by the vaccine, you pretty much are good, right? But again, remember, if that complexity of the challenge of making those vaccine is going to be tough. So this is a legend of whatever we discussed earlier. And some of the other slides, just a recap for you, because I know you like immunology a lot. So I don't have to teach you now and then. You're very good at picking up immunopathogenesis, how a body deals with the humoral immune response, cellular immune response, especially in terms of viral infection. And then again, which cells will come first, which cells will come later on. You pretty much know. And if you want to correlate the humoral and cellular response in terms of infection, you can see this is a time course. This basically would tell you, for example, the question you may ask, do I have a flu, the real swine flu? And remember, things are not easy. You don't want to go and test every one of them, every one of us, just because we suspect something. That's not a game over here. Even if you do, so you can see from here, one thing is that you look for viruses. The other thing is that you look for virus response in terms of antibody production. And you will see in all these sequences, especially from your book, it will show you that the virus-specific antibody, you can see from here, it basically comes about a week later because of the antibodies kicking in after a time. Okay? But again, symptoms come earlier. Viruses are shed in respiratory secretions, so they will pass it on to the people. That's why sometimes we have to make this decision whether we want to quarantine you or not, we want to put you in isolation or not. So that's the challenges that we normally have. But you can see as far as the pathogenesis is concerned, on the top you have to have a good cell-mediated immunity. You have to have interferon. Sometimes we give you gamma interferon to boost your immune system. And then again, if you want to look at the clinical disease, as I said over and over again, typical influenza. And normally for most of the viruses, especially for the flu, it's very debilitating. Viruses are debilitating anyway. Person would not recover from viruses less than a month if it's a severe kind of a viral infection. That's number one. Number two is the test. They're expensive, and you don't want to do it for everyone. Okay? As far as epidemiology is concerned, again, it depends upon which site are you looking at. You can look at the WHO. You can look at the CDC one, but what they want you to look at is that what is the strain type, where is it present, and how do we deal with it? And again, there are updates available which should be, and Those of you working in infectious diseases, I don't know whether it's mandatory or not. Is it mandatory for pharmacists to rotate in ID? No, I don't know. I don't know. Anyway, so, but I do have a elective that I teach for the current infectious diseases. Okay, so this basically, those of you, some of the slides are for those people who don't carry the book, and if you just want to go over the discussion points that I've been trying to kind of push through, and some of the things that should be there in your mind in terms of you yourself being protective against transmission of viruses. And if you understand the basic concept of enveloped versus a naked or non-envelope, you pretty much will really know what is going on. And then we talked about school children, they are together. And then we already talked about who is at risk. I mean, there was an initial slide for those of you who want to read it up. Pretty much the same. There are always risk factors that we discussed earlier as well. And the question you will have is that how do we control? So there's an antiviral drugs, there are tons of them. And there are vaccines available. But again, keep in mind that they could be killed or live vaccine depending upon what they are doing at that particular time. There are good tables in your book if you want to know them. But remember, sometimes most of you I see just get lost because it's too much information. What you need to do is that if you build up a solid base of understanding the viral diseases, everything is just the same. There's a little bit of difference. If you know what would an infection of upper respiratory tract do, it doesn't matter whether it's a viral, bacterial, you pretty much will have same pathogenesis apart from the causative agent. And you can see from here acute influenza virus in adults, children, and then complications. We already discussed them over and over again. And I believe personally that you don't need to basically know much about lab diagnosis, but the only thing that you have to keep in mind is that viruses cannot be cultured. Virus cannot be cultured. They can only be cultured if they were given those cells. Like we use monkey kidney cells. So this is not an easy test. And then again, we look for, if you look for antigen, we have ELISA tests. If you want to look at serology, that's where you look for the antibodies that you have produced. If you want to look at the strain and type of viruses, we have RT-PCR, some of the genetic studies that are there. And we already discussed in detail that the amantadine group and other analogs are going to affect a particular viruses because of the nature of their protective. We talked of Tamiflu is going to block the neuraminidase inhibitor. And then again, the other question, I remember I've been trying to convey to you. In viral infection, you have to be smart, you have to be quick, you have to do the diagnosis ASAP, because if you miss the window of 72 hours and you let the virus divide and spread, then it will be too little too late. That's why, especially even injectable drugs or oral drugs, acyclovir and other, they have to be initiated ASAP. So you don't want to sit on that, procrastinate, and delay the things. Because you can see there is an option for their 24 to 48 hours after the onset. So you have to act fast. Okay? So I think I'll stop here and then give you a 10